Hey everyone. Today I'm going to be going over Chapter 1, Section 1-2, Eukaryotic cells. And specifically I'll be reading through membrane-bound organelles. We'll go through the nucleus, mitochondria, and lysosomes. This can be found in your Kaplan Biology review book in Chapter 1. All right, so eukaryotic cells. The first major distinction we can make between living organisms is whether they are composed of prokaryotic or eukaryotic cells. And we'll be learning about prokaryotic cells a little bit later in the chapter. Eukaryotic organisms can be unicellular or multicellular. Eukaryotic cells contain a nucleus that is a true nucleus. It's enclosed in a membrane, but prokaryotic cells do not contain a nucleus. Although we will learn later in the chapter that they do include or contain a nucleoid region. All right, so the major organelles of a eukaryotic cell are the following: the nucleus, the mitochondria, lysosomes, endoplasmic reticulum, which includes smooth ER and rough ER, the Golgi apparatus, peroxisomes, and those are the big ones. There are some smaller ones, which include ribosomes, of course the cytosol, which is not an organelle, but that is the fluid in between the organelles. All right, let's jump right into membrane-bound organelles. Each cell has a cell membrane enclosing a semi-fluid cytosol in which the organelles are suspended. In eukaryotic cells, most organelles are membrane-bound, and this allows for the compartmentalization of functions. Membranes of eukaryotic cells consist of a phospholipid bilayer. Remember that, you guys, phospholipids in the membrane, something that we'll come to again and again when we learn about biology. So membranes of eukaryotic cells consist of a phospholipid bilayer. This membrane is unique in that its surfaces are hydrophilic, electrostatically interacting with the aqueous environments inside and outside of the cell, while its inner portion is hydrophobic, which helps it to provide a highly selective barrier between the interior of the cell and the external environment. So one way that I remember that is hydrophilic. I think hi Phil. It's a very open, electrostatically interactive layer, but the inner portion of the membrane is hydrophobic. Phobic, you can think of closed off, scared, much more selective. Okay, moving right along. The cytosol allows for the diffusion of the molecules throughout the cell. Within the nucleus, genetic material is encoded in the DNA, or deoxyribonucleic acid, which is organized into chromosomes. Eukaryotic cells reproduce by mitosis, which we'll learn about later. Mitosis allows for the formation of two identical daughter cells from one parent cell. All right, let's check out the membrane-bound organelle of a eukaryotic cell called the nucleus. As the control center of the cell, the nucleus is the most heavily tested organelle on the MCAT. It contains all the genetic material necessary for replication of the cell. The nucleus is surrounded by the nuclear membrane or envelope, which is a double membrane that maintains a nuclear environment separate and distinct from the cytoplasm. Nuclear pores in the nuclear membrane allow for selective two-way exchange of material between the cytoplasm and the nucleus. So just keep in mind that the envelope surrounding the nucleus has pores in it, and those pores allow for materials to transport in and out of the nucleus. The nuclear envelope creates two distinct environments within the cell because it separates the nucleus from the cytoplasm. This allows for the compartmentalization of transcription. Transcription is the formation, guys, of hnRNA from DNA, which is subsequently processed to form mRNA, and translation, which is the formation of a peptide from mRNA. The genetic material, or DNA, contains coding regions for genes. Linear DNA is wound around organizing proteins known as histones, and it's then further wound into linear strands called chromosomes. The location of DNA in the nucleus allows for the compartmentalization of DNA transcription, as we just discussed, separate from RNA translation. So let's put it this way. DNA transcription is happening inside the nucleus. RNA translation is happening outside the nucleus. Finally, there is a subsection of the nucleus called the nucleolus. That's where the ribosomal RNA, or rRNA, is synthesized. The nucleolus actually takes up approximately 25% of the volume of the entire nucleus, and if you saw it under a microscope, it looks like a dark spot. All right, we're moving on now to our second membrane-bound organelle in eukaryotic cells. It's the mitochondria. Think about the mitochondria as the power plant of the cell. They are very important for their function, which is metabolic functions. The mitochondria contain two layers, the outer and inner membranes. It's a good idea to look at a visual image. You can Google one or look right in your Kaplan Biology review book. But the mitochondria has these two layers, the outer and inner membranes. Picture it this way. The outer membrane serves as a barrier between the cytosol and the inner environment of the mitochondria. The inner membrane is thrown into these numerous enfoldings called cristae. It looks kind of like a maze if you saw it under a microscope or in a digitalized or illustrated image. So these numerous enfoldings called cristae, that's C-R-I-S-T-A-E, contain the molecules and enzymes necessary for the electron transport chain. Just put a little flag on that, electron transport chain. We'll learn about that another time. The cristae are highly convoluted structures that increase the surface area available for electron transport chain enzymes. The space between the inner and outer membranes is called the intermembrane space, whereas the space inside the inner membrane, or inside where the cristae are, is called the matrix. Now, this is important. The pumping of protons from the mitochondrial matrix to the intermembrane space establishes what's called the proton motive force. Why is proton motive force important? Well, it's central to the whole function of the mitochondria as a power plant or energy producer. Ultimately, these protons flow through ATP synthase to generate ATP during oxidative phosphorylation. Mitochondria are different from other parts of the cell in that they are semi-autonomous. They contain some of their own genes and replicate independent of the nucleus via binary fission. As such, they are paradigmatic examples of cytoplasmic or extranuclear inheritance. Bingo, just put a little bookmark on that term. Extranuclear inheritance. It is the transmission of genetic material independent of the nucleus. So mitochondria are great examples of this extranuclear inheritance, also called cytoplasmic inheritance. It's when we transmit genetic material in an organelle independent from the nucleus. Mitochondria are thought to have evolved from an anaerobic prokaryote, engulfing an aerobic prokaryote and establishing a symbiotic relationship. If you're not sure what anaerobic and aerobic prokaryotes are, don't worry. We're going to get to that very soon in this actual chapter in just a few moments. Here's a real-world example. The serial endosymbiosis theory, I'll say that again, the serial endosymbiosis theory, or endosymbiosis theory, attempts to explain the formation of some of the membrane-bound organelles. It puts forward that these organelles formed by engulfing one prokaryote and another, and those two prokaryotes established a symbiotic relationship. So in addition to mitochondria, chloroplasts in plant cells, and organelles of motility like flagella are believed to have evolved through this process. That's, again, the serial endosymbiosis theory. All right, we're moving right along. One more thing I want you to know about mitochondria. In addition to keeping the cell alive by providing energy, the mitochondria are also capable of killing the cell through a process called apoptosis. And now, moving on to lysosomes. Lysosomes are membrane-bound structures containing hydrolytic enzymes that are capable of breaking down many different substrates, including substances that are ingested by endocytosis and cellular waste products. I like to think of lysosomes as Lysol. It's a cleaning agent. The lysosomal membrane sequesters these enzymes to prevent damage to the cell. However, release of these enzymes, these cleaning agents, if you will, can occur in a process known as autolysis. Like mitochondria, when lysosomes release their hydrolytic enzymes, it results in cell death or apoptosis. In this case, the released enzymes directly lead to the degradation of cellular components.